Alright, so this video is going to focus on reviewing for the domain test over all of the genetic units. So this covers our units 4, 5, and 6. And when I go through this, I'm going to be hitting the major points and kind of some words to help remember the different information. So starting out, we're going to start with SB1B, which is develop and use models to explain the role of cellular reproduction, including binary fission, mitosis, and meiosis, in maintaining genetic continuity. So remember, continuity means is the noun form of continuous, which means to keep going. So first thing that you had to do was label the different descriptions about the parts. So mitosis has 5 parts: prophase, metaphase, anaphase, telophase, or if you can remember the acronym PMAT, which stands for each of them. Interphase is going to be before them, so when the cell is not dividing, and then cytokinesis happens at the same time as telophase, and cytokinesis is when the cell actually splits. So the second question asks you to label the different images, so you need to be familiar with these. So the first picture here is going to be anaphase, because the way you can remember that is that it's going to be separating the chromosomes and they're going to be pulled away. The second picture is metaphase because they're lining up in the middle. The third picture is prophase. Prophase means before, so you're before the cell is starting the movement of chromosomes, so we're removing the nucleus. The spindle fibers, which are those long threads, are forming. And then the final picture is telophase. So this is like, think of tele at the end. And so the nucleus is reforming and the cell looks like it's about to split. Then from there, we're moving on to the different kind of definitions and terms. So haploid and diploid are terms we use to describe the results of either mitosis or meiosis, the different cell divisions. So diploid, think di-B2, and for haploid, think of hap sounding like half. So di is 2, and hap is going to be like a half. So with that being said, you have 2 copies with a diploid cell of each chromosome. So for humans, that's going to be 46 total chromosomes. For haploid, you have 1 copy, so 23 in humans. These are going to be, diploid cells are going to be your body or somatic cells, whereas haploid cells are going to be gametes, your sex cells, your egg and sperm. Diploid cells come as a result of mitosis or binary fission, and haploid comes from meiosis. And so that leads us into the difference between them. Mitosis makes body cells. It's asexual reproduction. They create 2 cells because we only divide once. We said they create diploid cells, and it does not have crossing over. So as a reminder, crossing over is where the pair of chromosomes swaps information. Meiosis is going to make the gametes. This is sexual reproduction. It creates 4 cells. They are haploid, and it does involve crossing over, so we create genetic variation. So in mitosis, the number of chromosomes is the same. The DNA is the same. It's a clone of it. Binary fission is the same as mitosis, but it's what is found in bacteria and single cell organisms. And then meiosis is where the chromosome number is halved when the offspring are made. Alright, so then we move into SB2A, construct an explanation of how the structures of DNA and RNA lead to the expression of information within the cell via the process of replication, transcription, translation. So first thing about DNA is that it is double-stranded. You need to make sure that you understand that the strands of DNA are held together by hydrogen bonds. And the biggest thing here really in terms of the DNA information is knowing the differences between DNA and RNA. So this table is really, really important. So we start with there are 4 different things that we can compare in DNA and RNA. First is the sugar. This is what makes them up, and this comes from the name. So D in DNA stands for deoxy or deoxyribose, was a sugar. For RNA, it starts with R, so it's going to be ribose. DNA is a double helix, it's double-stranded, so that's its shape. For RNA, it's single-stranded. There are 4 bases in each one. In DNA, it's A and T, which go together, and then G and C go together. In RNA, it's A and U, and G and C. DNA is found only in the nucleus, but because there are different types of RNA, which we'll get into in a moment, they can be found in the nucleus, and they are also found in the cytoplasm. So they move between them. The molecules of DNA and RNA work together to form what we call the central dogma. And this is how the information flows from the information in DNA to make proteins. So this is the kind of the result of protein synthesis. Everything starts with DNA. DNA can copy itself, which is called DNA replication, which takes place in the nucleus. So I'm kind of combining this question here, this diagram here, and this chart. So DNA replication's purpose is to copy DNA. It takes place in the nucleus because that's where the DNA is. And it uses a molecule known as DNA polymerase, which is what actually copies the DNA. If I'm going to be making a protein, the first step is called transcription. Transcription takes the DNA and copies it into RNA. Transcribe means to copy. So if you think of transcribing, I am not sure what happened there. Okay, so if we think of transcribing, so to transcribe is to copy. And so we're copying the information DNA into RNA. It's going to take place in the nucleus because that's where the DNA is. This time it's using RNA polymerase because RNA is being made. We're going to make the mRNA and obviously use the DNA. For translation, to translate something is to change the language. So think about translating between English and Spanish. And so DNA and RNA are nucleic acids. That's one macromolecule. Protein is another. It has amino acids. So in translation, we're making the protein. This takes place in the ribosome. The ribosomes can be on the rough ER or they can be in the cytoplasm. But we focus on that is the location, is the ribosome of translation. So you have mRNA and you have tRNA, and we'll talk about what those are in a second. But the purpose of translation is we take that information from the DNA and RNA and make, specifically the RNA, and translate it into the protein, the amino acid sequence. Okay? And when we do DNA replication, we call it semi-conservative because you end up with one original strand and one new strand. Now, in the RNA, there are 3 types, 2 of which we're going to carry about. First is the messenger RNA. That is what's created in transcription. It has the information from the DNA and it carries it to the ribosome. It's the recipe to make the protein. The tRNA is another key component, and that is what brings the amino acid. It's the thing that's bringing the supplies to make the protein. So the tRNA will bring the amino acids to form, and those amino acids will come together to make the protein sequence. You also need to be able to use the base pairing to figure out the different sequences. So what is given in black here is the original sequence. And so as a reminder, when we're doing this DNA sequencing, A pairs with T and G pairs with C for DNA. Okay, this is specifically DNA. Okay? In RNA, A and U go together, and G and C go together. Okay, in RNA. So, ATG in DNA will be TAC. CCA becomes GGT, and GTC becomes GAG, ATC becomes TAG. The same idea when we're going from DNA to RNA. So if I were to put a T in the DNA, I change that to U. So ATG becomes UAC. So you still pair the A and T together in RNA because those go together. The only thing is when you're doing the RNA sequence, you replace what you would normally put with a T with a U. So if I compare the first bullet point here to the third bullet point, so the opposite DNA sequence and the mRNA sequence, they're the same except for wherever there's a T, there's a U down here. And then we use the codon chart, which will be provided on the test for you, to figure out the amino acid sequence. So this is tie, glide, glue, and then stop. So on the codon sequence, I don't have an example of this here, but I'm going to draw it off to the side here, if it will let me. Nope, it's not going to let me. So let me erase this that I've written, because you guys can have written that down earlier. Let me erase all this so I can kind of sketch out the codon chart. And if needed, your teacher can review this with you. Okay, so the codon chart is going to be a box. Go on. It's going to be a chart, so it's going to have 3 sides. Okay, this is a little tricky here. Okay. All right. So the way that it's going to work is your first base is going to be over here. So we would have A, T, G, and U. Across the top would be your second base. So A, T, G, and U. And then on the right, you would have your third base. And this is where it gets a little weird. So within, you would do your row and your column, and then within this row and column, if we imagine it going down here, there would be 4 options. And then on the side here would be the different bases. This is a different order, but basically you would clue in where the 3 come together. So you would find, so like if I'm doing UAC, I would go the row for U, I would do the column for A, and then in that grid, I would find the area that's C. So that's where I would be. I just realized I wrote the wrong thing here. This should be C. So hopefully that makes a little bit of sense. It's kind of hard to envision without the actual codon chart, but the codon chart will be provided for you. Just remember that it's first on the left, second on the top, and third on the right. All right, so now we're going to move into mutations. With the mutation stuff, you really just need to kind of know the definition. So this is SB2B. It says construct an argument based on evidence to support the claim that inheritable genetic variations may result from new genetic combinations through meiosis, so crossing over and non-disjunction, non-lethal errors occurring during replication, so insertions, deletions, substitutions, and or heritable mutations caused by environmental factors, radiation, chemicals, and viruses. So first of all, crossing over, we talked about this earlier. This is where segments of a pair of chromosomes are going to switch, and this happens in meiosis to create differences or variability. If we have mutations within the DNA, there's two types. The first is point mutation, where we change a single base. So if CGT, CCA is my original sequence up here. If you will see here, I've changed the G to a C in that the end of the first three letters. So that's a point mutation. I'm changing one letter. Frame shift is where I've either added or deleted. So if you look at that second sequence here, I deleted one of the C's from the third group of three. So the two types of mutations that you can happen within DNA is a point mutation where you just have a single change, or a frame shift where I add or delete. For a chromosome, I have non-disjunction. This is where Down syndrome comes in, where you would get too many or not enough. So what you see here on the far left is going through meiosis, you get one copy of every chromosome. But in an abnormal situation, it cannot split properly. So one side is correct, but the other side isn't. So like at the second division, it doesn't separate correctly, so you can see here that second cell has 3 where the first one has 1. This is considered non-disjunction. It can be not enough, so you only have 1 copy, or too many where you have 3. This is for humans. And then you can have the same thing happen, but at the first division here, and that would be where 2 of them would have 1 copy, 2 of them would have 3 copies. And so that's non-disjunction. A good example, like I said, is Down syndrome, and we'll talk more about that in a minute. All right, next is SB2C, ask questions to gather and communicate information about the use and ethical considerations of biotechnologies in forensics, medicine, and agriculture. With this, you really just need to understand what each kind of technology does, and then you need to be able to identify or interpret a DNA fingerprint. So gene therapy, which is that first definition, is essentially trying to fix a gene by replacing it. Genetic engineering is different. We are trying to change the DNA to give new traits. So that is take away something, add something. Gene therapy you can almost see as a specific type of genetic engineering. So gene therapy, we're trying to fix something, whereas with genetic engineering, we're just trying to change something. Gene sequencing is just what the name says. We're trying to figure out the order of the letters. Cloning is where we make an identical copy. So mitosis is an example of cloning that happens naturally. DNA fingerprinting is, we'll look at in a second, but it's basically a pattern that's created. And then a technique that creates lots and lots of copies of a specific section of DNA is called PCR. And so you can see down here, the question says, Lieutenant Russ is investigating a murder scene. The felon was scratched by his victim, and some of his skin cells were found under the victim's fingernails. A DNA test was performed, so this is a DNA fingerprint. To which of the suspects do the skin cells collected from under the fingernail of the victim belong? So the felon is here on the far left, and then we have 1, 2, and 3 suspects. And the way you interpret this is the one where the bands match, that's your suspect. So this is one instance of where DNA fingerprints can be used. Another instance would be for paternity, to figure out whether it was the father. So think daytime talk shows of who's the father. This is how they do it. And again, it's a pattern matching. You're matching the reference, which typically is in that first column, to one of the others. All right, so now we're going to be getting into more recent stuff. So SB3A is use Mendel's laws, segregation and independent assortment, to ask questions and define problems that explain the role of meiosis and reproductive variability. So let's review the 3 laws that Mendel had. The first is law of independent assortment. So genes are inherited independent of each other in meiosis. So what this means is your genes for your height and the genes for your hair color do not influence each other. So you can be really tall and have brown hair, or you can be really short and have blonde hair. So that means that they're not influencing each other. They're not inherited together to say that if you're tall, you're going to have brown hair. Alleles, the law of segregation says alleles are separated when gametes are formed in meiosis. So every person, when an egg or sperm are created, the starting cell has 2 copies of each allele because we have 2 chromosomes, one from mom, one from dad. So as the cell divides during meiosis, those copies are going to be separated. So at the end, we get haploid cells, so there'd be 1 copy of each chromosome. And then the rule of dominance, sometimes called the law of dominance, just says that one allele will overpower the other and become more common. So the dominant allele is more common, and you only need one version of it to show the trait, whereas with the recessive allele, it's less common and will be masked or overtaken by the dominant, and you need 2 copies of the recessive to show the trait. Genotype, think genes. It's the letters that we use, the capital, lowercase, that type of thing. Phenotype, think physical. That's the physical expression of that. What do we see or what trait shows up? And then if we're using rabbits here, we have 3 forms of the genotype, or 3 versions of the genotypes. We have homozygous dominant. Homo means same. So that means they're both dominant. 2 capital letters, and it's going to be, in this case, brown, because brown is said to be dominant. You just have to have 1 capital letter in order to show the trait. Heterozygous. Hetero means different. So if we go here, as I'm fighting with my computer, doesn't like me having to write. Okay, so again, homo equals same, and hetero equals different. And so for heterozygous, you are going to have 1 of each letter. And even though it shows it has one of the even though it has a recessive version, it's going to show the dominant trait. So that's different. So in this case, it's brown. Homozygous recessive, you have 2 little letters, and they're going to show the recessive trait. So here it's lowercase, here you have 1 big and 1 little, and then here you have 2 big. So again, homo means same, hetero means different. Just think of those. All right, let's get into the Punnett square. So SB3B says use mathematical models to predict and explain patterns of inheritance. So mathematical models are going to be your Punnett squares. We have 2 types. The monohybrid. This is the one that you've done a bunch in class. This is the one you need to be really familiar with. So it says here, if a heterozygous fire-breathing dragon is crossed with one that does not breathe fire, which portion of the offspring will be fire breathers? Fire breathing is dominant to non-fire breathing. So hetero means different. So one parent would be capital F, lowercase f, and you put them at the top here. The other parent is not breathing, so they are recessive, so 2 little f's, and you put them on the left here. And then you just match the column and the row. So a big F, little f here, big F, little f. This is 2 little f's, and this should be a little f there also. So again, the big letter comes first, you just match the row and the column. And so in this case, it wants to know how many will be fire breathers, so that's at least a big capital because it's dominant, and that's 50%. This is known as a monohybrid cross because it's 1 trait. A dihybrid cross is much more complicated. It's 2 traits, and so you can see the box is bigger. You'll have to be able to do 2 things with a dihybrid. Fill in empty boxes, which we'll talk about in just a second, and look at the genotypes and count up the number. So it says a male rabbit with a genotype of homozygous dominant, homozygous recessive, is crossed with a female rabbit with a genotype of homozygous recessive and heterozygous. The square is set up below where capital G is gray, lowercase g is white, capital B is black, and lowercase b is red. So the male rabbit is gray and red. The female rabbit is white and black. So complete the squares. So literally all you do is, like with the monohybrid here, is you just do the row and the column, and you match the letters together with the larger letter going first. So this is capital G lowercase g, so I put that, capital B lowercase b. Here in the second row is going to be capital G lowercase g, and then 2 lowercase b's. Does it matter if G or B comes first? No, but that's how it's written. Same thing down here as the first row, and then again we repeat the fourth row. Then for part A and B, I look at the letters. So gray and black are both dominant, so they only have to have one of each letter. So I'm going to count the boxes that have one capital B and one capital G. So this one does, this one does, this one does, this one does. We all have capital G's in the second row, but we have lowercase b, so that doesn't count. And then all of them in the third row have a capital B and a capital G. So that's going to be 8 total, and there's 16 boxes, so I would just put 8 here for part A. The second question says white fur and black eyes. So white fur is going to be 2 little g's, and black eyes is going to be capital B. Well, none of the boxes have 2 little g's, so the answer here is 0. And you can have 0 for these. So again, with a dihybrid, there's 2 traits. You need to be able to fill in missing blocks, like we've talked about, and be able to look at the letters and kind of count up how many have certain traits. For the different inheritance, there were 5 types we talked about. Sex-linked is common in males. They are going to be recessive. Polygenic, poly means many, genic means gene, many genes. The way this is going to show up is it's going to have every shade. So the example we're using is blue or yellow scales in lizards. So you would have all sorts of the shades starting with yellow, going into green, going into blue. That's polygenic. Think height, skin tone, hair color, eye color, things like that. Incomplete dominance, it's where you have 2 forms, and then the middle form, the heterozygous, is a mix. So if we have blue and yellow, those mix together make green. Codominant is where both show up in the middle form. So we would get yellow and blue spotted, striped, checkered, there's different ways to say it. And then complete dominance is what Mendel had. Either you have blue or you have yellow. And so really with these, you just need to know how to tell. That's the big thing here, is how to tell which inheritance or which type it is based on a description. So in this, there's a different example. So like male lizards have the recessive color yellow. So being able to use those and pick those out. All right, a pedigree. For a pedigree, this is a way to trace an inheritance in humans. Humans are kind of hard to study. And so a pedigree shows a family, basically family tree and who has what trait. And there's 2 kind of categories you can have. You can have autosomal or sex-linked, and then you have dominant or recessive. So autosomal, the ones filled in are going to be about equal male and female. Remember, males are squares, females are circles. You can have more females with autosomal, but the big thing is it's not all males, which is sex-linked. Dominant, most people are going to have it. It's going to be in every generation. Recessive, very few people are going to have it, and it's going to be skipping generations. So if we look at this pedigree here, it's for the inheritance of sickle cell anemia. We look at the ones shaded in. We have 2 males and 1 female. That's about equal. We do have slightly more males, but that's still about equal, so it'd be autosomal. For whether it's dominant or recessive, it skips generations. There's only 3 filled in out of all of these people, and that's recessive. So it's autosomal recessive. What that means is anybody who's filled in will have little letters because they are recessive. Anybody who's not filled in here will have at least 1 capital letter. So that leads us into question B. Why can you say that generation 2, individual 3 and 4 are heterozygous? So the numbers here, the Roman numerals 1, 2, and 3, are the rows, which are the generations. and then the individuals are just numbered 1 through however many. So for 3 and 4, we are looking at this couple here kind of in the middle on the left, right here. This is the couple we're looking at. The reason we can say that those are heterozygous is because this is a recessive trait. They have children that have it. So both parents have to have the trait and or have a lowercase letter in order for their children to get it. So that's why we can say they're heterozygous. Let's see. Do I have this space? Let me see if I can draw a Punnett square real quick. Hopefully we can show that. So if we're going to use, we'll just use A to do this, if the computer will cooperate with me. Okay. I'm going to use a space down here next to the karyotype. So actually, no, I can do it up here. Okay, so I draw my box. Okay, so if both parents are heterozygous, big A, little a, you'll have one box with dominant for both. You'll have 2 boxes where it's heterozygous, and then 1 box where it's homozygous recessive. And so they have to be heterozygous because 2 of the children are basically this lower box here. And so that's why they're heterozygous. Alright, moving on to the karyotype. So a karyotype is this picture here. It shows all the chromosomes that an individual has. And it's a way that we can pick out if there's anything wrong with chromosomes, like physically, like we have too many, there's an issue with, there's a deletion, there's a removal, things like that. We can also tell the sex. So based on this karyotype, the main issue that you can see is that there's extra chromosomes and there's 3 spots. You have 3 at 8, you have 3 at 19, and you have 3 at 22. Does this cause a specific illness? Not that I know of. But we can also look at the X and the Y. In this case, there's 1 X and there's 1 Y, so that makes it male. The X and Y markers here, these letters, will always show up. If there's not something above the Y, if there's 2 things here at the X, that will be female. So just don't be confused if the Y is there. The Y is always there. Whether something's above the Y tells you if it's male or not. And there's a specific case that we've talked about called Down syndrome. Down syndrome is 3 copies of the 21st. So that's a very, very common one that is used as an example of what we call non-disjunction, or something you can see on a karyotype. All right. Finally, to wrap up, the last standard is SB3C, construct an argument to support a claim about the relative advantages and disadvantages of sexual and asexual reproduction. We've talked about this a little bit more so when we were talking about mitosis and meiosis. Asexual reproduction is mitosis. You're creating identical offsprings. The advantage of this, it's quick. You only need 1 parent. Disadvantages, it is identical offspring or daughter cells, and so you don't get the genetic differences. You don't get that variability. For sexual reproduction, it's the opposite. Meiosis is involved with sexual reproduction, and the parents are different. The offspring are different from the parents. So the advantage is that you get the variation. And the disadvantage is that it requires 2 parents, more resources, more time. And so if you look at asexual and sexual, they're opposite to each other. Also, not only in terms of the process, like whether it's mitosis or meiosis, but also the advantages of one or the disadvantage of the other. So the fact that asexual is fast, the disadvantage of sexual is that it's slow. So it's like they kind of are opposites of each other. Hopefully this has helped kind of hit the high points and put it in some more simple